Hello, my name is Harshit Gupta,
I'm Abhinav Menon.
and the aim of our project is to use computer simulations to implement Hodgkin-Huxley models of various cells. These cells are drawn from various regions of the brain, like the visual and somatosensory cortices, and from various animals like ferrets, rats, guinea pigs, etc. We also plot the voltages and the various constituent ion currents along with their gating variables. After that, we analyze the periodicity of these currents under a repetitive spiking behavior.
So I'll be presenting the code that we have used for our project. We have implemented models for 4 different types of neurons: fast spiking, intrinsically bursting, low threshold spiking, and regular spiking. We've used the Brian library of Python and implemented different models for each class of neurons. For example, we have here the fast spiking class of neurons, for which we have implemented the ferret visual cortex and rat somatosensory cortex models. And yeah, so this class of cells includes the sodium and potassium currents as well as a leak current. The leak current is constant and common to all the cell types. The sodium and potassium currents are controlled by the gating variables m, and h. So these are the equations. As you can see, m and h are controlled by the differential equations that we have here. And similarly, the rat somatosensory cortex, which has an additional slow non-inactivating potassium current called IM, which is controlled by a gating variable p. And yeah, so this is the fast spiking model. Similarly, we have the intrinsically bursting model, which includes a sodium current, a potassium current, a slow non-inactivating potassium current, and a calcium current, as well as the leak current that is common to all. The calcium current is controlled by 2 gating variables q and r, which have more or less similar form to m and h, which we have seen before. And yeah, so the cat visual cortex and the guinea pig somatosensory cortex are the cells that come under the intrinsically bursting class. Then we have the low threshold spiking class, which includes sodium, potassium, slow non-inactivating potassium, and a different kind of calcium current called IT, which is controlled by gating variables S and U. And yeah, this includes the cat association cortex and the rat somatosensory cortex cells, which both include the current types I mentioned just now, as well as a leak current. And finally, we have regular spiking cells, which includes sodium, potassium, and slow non-inactivating potassium currents, just the 3 kinds, which are modeled by the gating variables that we have seen till now: M, H, N, and P. And these are the differential equations that control the values of these gating variables. This class includes the ferret visual cortex and rat somatosensory cortex cells. So as Harshit mentioned, we can see that you can see that we have implemented various types of models for modeling different regions of the brain from different animals, following all these 4 cell types. And now Harshit will explain how we plotted the various variables.
So regarding the plotting of the various variables, what we did was we first plotted the voltages and we restricted the voltages till the time 2 spikes are generated. The reason for doing so was so that we can analyze the current and the other important gating variables much better. So after restricting them to 2 spikes, we then modeled all the current components and the gating variables along with the other components such as tau, which affects the gating variables. And yeah, we can see the plots for each of these things. So this is how we went about plotting it, and all our plots we can see it in this particular folder for each of the different cell types and then the animal types. Yeah.
So yes, so as Harshit mentioned, we plotted all the variables in a time frame corresponding to 2 to 3 spikes, and this specifically belongs to the limiting cycle behavior of the cell, which exhibits repetitive spiking. So if we extend the time period, we can see that under these conditions, the models present an arbitrary number of spikes, and the exact parameters used in these plots can be found in the code, which Harshit just showed. So for example, here we start with the default voltage of -75 millivolts and an injected current of 0.7 nanoamperes. So yeah, so for lack of space in our report, we have presented the plots for a single model for each cell type. For example, the ferret visual cortex in the case of regular spiking, the rat somatosensory cortex in the case of fast spiking cells, and so on. And yeah, so this is so yeah, and so here in fact you can see here the diversity of animals from which the cells have been taken: cats, guinea pigs, ferrets, and rats. And yeah, and now and these are the equations that each of the that we just saw implemented in Python. These are the corresponding mathematical forms of the constituent currents that form part of the models. So yes, so the various observations we've made from these plots are, firstly, that the current is voltage spiking is mostly dominated by the sodium current. So we can see that the sodium spikes correspond exactly to the current spikes, sorry, to the voltage spikes, and happen at exactly the same time. And the other currents, potassium and calcium mainly, never rise above 0. So they have a kind of inhibitory effect on the spiking. And yeah, also the component currents model the increase and decrease in the peak of the spike. So many of the models show a different peak in the second spike from the first spike. For example, in fast spiking, the peak has fallen, and in intrinsic bursting, the peak has risen. And this is modeled by certain specific gating variables for each of the currents. So for example, in the case of sodium, we have the H gating variable, which has decreased in value, and that has shown that models the synaptic depression in this case. And similarly, in the case of sodium, the H value has risen in the second spike for intrinsically bursting cells, so that models the synaptic facilitation. So we can see specifically that the M gating variable has absolutely no effect is not affected by the synaptic depression or facilitation. It remains consistently periodic, right? And yeah, so each of the constituent currents model this peak or fall. So for example, we can see here that even the potassium current modeled a smaller peak, and so did the slow non-inactivating potassium current. Yeah, another important observation we made is that the second assumption that underlies the Fizio-Nagumo model, a simplification of the Hodgkin-Huxley model, was is supported by these plots. So for the gating variables H and N can be compressed into a single gating variable, since their dynamics are very similar. And yeah, this is borne out by our plots. For example, we see here that H stays close to 1 as long as N stays close to 0, and they both rise at almost exactly the same instant, and again fall in a manner mirroring one another, so that they can be modeled by a single gating variable W, as posited by the FHN model, which is 1 minus H equal to AN. Yeah, and that is another important observation we made. And another observation we have was that the gating variable P, which is most clearly visible here, is monotonically increasing rather than periodic, as opposed to all the other gating variables. And so it has a great effect on the depression of the spike in the case of the slow non-inactivating potassium current. Yeah. So these are our main observations. And so these observations have contributed to our understanding of the Hodgkin-Huxley model and the Fizio-Nagumo model, specifically by giving us some insights into how the models are biologically realistic in terms of modeling constituent currents and the openness of ion gating channels ion channels through gating variables, and how the assumptions underlying the FHN model are in fact statistically valid, especially the second assumption that compresses two gating variables into a single one. So, thank you.